Good Lord, that's loud.
Good afternoon, everyone. Let's go ahead and get as always, this is just kind of here before the yes, we do have lecture on Wednesday, but and it will directly connect to the test, so don't and we'll be talking about different during that lecture, we'll be talking about different projects, how they work, why they become kind of a big 10 years how you get involved more if you're interested. And then we'll talk about how the citizen science project that we ran, where we went out and hiked all those trails that is due tonight. Talk about some of the cool critters we found. Basically, for context so far, we've doubled the number of species observed on campus. So before this whole iNaturalist thing, we're double the amount that we had previously observed on campus. Just from the work we all done in our campus here, that's really cool to me. We'll talk about some of the two male cohorts kill each other. Yeah, so just for context, tortoises are really meat. Well, that's what Wednesday's lecture will be about. More than likely, we'll be getting out here about in like 30 minutes. So if you have any extra questions, I'll answer them. All right. So let's just go ahead and get started with our normal kind of rundown of things. Quiz 11, which is the last quiz, is Sunday, November 7th at 11. And as always, I really highly encourage y'all to take it before the exam. This one will probably focus on those last two lectures from last week, and prepare you for what's coming up. Especially a lot of the questions you're going to be practicing your knowledge. What groups have certain things they want to know. Remember, it's open notes, so use that opportunity then to kind of use that as a study guide for you. As I've already kind of alluded to multiple times here, your third exam is this Friday at 5 times. Again, we're offering that dual style. I know it's not perfect, but we're working on it. So take it online if you can. It'll be roughly the exact same amount of questions, exact same amount of time to take it. But again, see that response browser. So if you have issues with that, I highly I'm really trying to get a head count on it this time. Be ready. So just let me know ahead of time and shoot me. For those of you that are taking it at the Testing Accommodation Center, the big deal with that is don't worry about a Scantron or anything like that. I'm just going to I'd rather just be able to see it easier on the sheet than it is to deal with the Scantron. So that's fine by me. Questions or anything like that overall? Let me know. But again, I haven't quite figured this out yet. They're pretty similar to the questions that are on the quizzes, just tweaking about version A, asking you what kind of things are translated in the root. And the quiz opposite, so what kind of things are transported or created in step. That makes sense? All right, does anybody have any questions before we go on from that? Perfect. But yeah, as always, don't hesitate to reach out if you have any questions or need anything. If something comes up and you're not able to make that time, and again, just let me know. Again, remember, I do need a proof of it absence or something, but especially if you need ahead of time, I have a lot of possibilities. Better because I can actually do a lot of things, and it's like 2 days after instead of my option. All right. So along with our third exam, we do have an extra credit opportunity. Again, last time this is only 5%. So if you can which did make a difference in exams. I saw a number go. Keep in mind with this one is that you can observations. So if you observe, animals like your animals were like insects, you obviously have to go find predators, right? It's not that big a deal. I've given some instructions on where you try to find dogs. Specific questions, I'm always happy to answer. Birds ask questions. Imagine that birds around the world. On campus, there's like 3 or 4 birds.
I'm always calling at once. So as always, if you have any specific questions.
Are we allowed to see like
for the connecting with Byron and the extra credit. Walk, which is not technically also part of the true arboretum, but still they have. I think making like is we covered the arboretum. So covering some of like clear too.
Yes.
I don't know if those I have to sign it instead of having to do like secret a video of the bird calling.
So for this extra credit, either the sound observation or the photo observation is fine by me. The only thing you can't do is just have like a blank picture or something like that. We will double check most of them. Obviously, we can't double check everything, so there may be a couple things that slip underneath our radar, but between Katie and I, we're trying to get through these and make sure y'all are kind of being held to that standard.
It's kind of fun, right? All the different wildflowers and stuff.
It's neat. But yeah, anything is fair game as long as it's accepted by iNaturalist, as far as I'm concerned. All right. And the one last thing, of course.
Connecting biology science. Keep in mind that it's really good. Make sure yours matches the sample submission. I don't match, you're gonna lose points. And I try to make it as like standardized, objective as possible. For instance, if you just observations instead of posting a screenshot from iNaturalist showing you actually observe these things up there, that's only one time credit for that, like 5 points. So it's a lot. So it's a lot of just little things, and I promise you all these things are I'm taking that information
Because it's important to know when you're out walking along this trail, one, where you're at, see what time of day it is, what kind of weather, because all of that stuff influences what kinds of things you're going to see. And that's important when we're trying to figure out what kind of weather conditions. So, as always, if you have any questions about that assignment, please don't hesitate to email me or email Katie.
Both of us have practiced this assignment. In fact, I've done so I would have actually observed. Actually walking part. Myself. Necessarily not necessarily because of coyotes.
And along those lines too,
I know we kind of joke about this, but to be honest with you, there have been quite a few reportings of raccoons attacking people on campus.
Basically, what we believe has happened.
Is, during COVID, they kind of got really protective of very specific spots because there were people around and there was always people around. As a result, if you go near some of those, you may be run off. And to be honest with you, there's no guarantee that those raccoons don't have things in front of you. Even rabies, the treatment for it is like 10 to 20 shots that go straight into the abdomen with like the thickest needle. It is absolutely miserable. You do not want to fuck that. Like so. Just want to make it, and along those lines too, there are rattlesnakes on campus. There are cotton bats on campus. It's keep in mind too. So for context, when it comes to venomous bites, the most likely to get bit 18 to 25-year-old males.
Yeah, don't be stupid, please.
And I can say,
Don't be stupid. But again, if you have any questions, please just let me know. I'm happy to help out however I possibly can. This assignment's supposed to be an easy, quick, go walk outside, observe things, come back inside, write things down, and then turn it in. Don't overcomplicate it. Now, I do want to point something out real quick, though.
this is what your things should look like. So my max house, where it's probably better to see the screenshot of the actually go off on time and that's where you can explore function will either version or on that version. Technically, it'll be the zoom a lot just where your stuff is. Leave your name, your number of observations, your plants, the kind of observation, what trend you are, what conditions, and then that screenshot of my naturalist with that identification. If you only identify to animals or to plants, that's not correct. You have to go down to those four categories of gymnosperm, angiosperm, brontocyte, vascular, non-vascular plants. Same thing with animals. Other than vertebrates, you're only going to do like kind of work. You're going to find part of and what they're doing, when it comes to vertebrates, you don't know the difference between a bird, a reptile, and a living and an by this point. If you haven't already heard it, I heard it, right? So just make sure you're filling this out. If you do all of this 100%, that's 15% area total between. And again, that's all you have to do. Yeah. So the map is important. So the reason why I'm asking for that is basically what it's doing is it's plotting where you observe specific things, because obviously you can see all of y'all together, but it's a lot harder to like pinpoint one individual person. Yes.
When I was walking the trails, I dropped in some Google Maps with the opinion that.
Yeah, that's fine too.
Either way you do it is fine, as long as you have a map with where you observe things.
I heard somebody else.
So one of the things I did want to mention is that while the overall average for the people that have submitted,
there were people that didn't post a screenshot. I think I was clear enough when I made this instructions that so I went ahead and reopened it and allowed you to be able to submit it a second time. That issue, feel free to resubmit or feel free to resubmit. Don't worry. Honestly, if you have questions like that, don't hesitate to reach out. Here's a resource. Ultimately, my main goal of this class is I want you to hear a little bit more about my data. That's fine. I'm trying to do it. And happy to kind of talk this through. Don't hesitate to reach out.
All right. Any other questions before we get started properly? Cool. So today we're going to just do a quick review through the findings that y'all kind of struggled with.
We'll do these 10 slides, so we'll go through those rather quickly, and then as y'all have questions in here. So let's go ahead and get started.
Now, the first thing I want to point out is remember that we talked about how biologists determining the structure of DNA in 1955 determined both the chemistry and how it's physically structured, right? To get that, we finally figured out how that double helix looks and what DNA actually looks like.
Now, there was some confusion here, and I'll argue that some of the my part I should have been more clear. But there are 3 people, and there's a really good story on it, and there's a lot more outlets that go. But ultimately, what happened is independently, Bob Franklin and Watson and Crick were working to try to understand what this molecule was. Now, as a result of their working on each other, from each other, they heard about each other's work, the conferences, and policy of their scientists, the conference in the general field, and just talk about their research. I'm sure that these 3 about all the shit they were doing next. And what ended up happening is that Rosalind Franklin shared some of her expert images to figure out what DNA. But that wasn't quite to the resolution yet. So all 3 of them worked to improve the techniques a little bit and get those polished images. And then Watson and Crick just totally ignored her. Really shitty thing to do. Unfortunately, technically, all 3 of these individuals were involved in that discovery of that double helix. So yes, it really needs to be said that Russell Franklin needs to get more credit. And part of the reason why she doesn't have a Nobel laureate, she actually has to be really young. And unfortunately, one of the rules with being a Nobel Prize winner is that you have to be alive to get it. Actually, I think in her early 30s, less than 5 years after they discovered this. However, they didn't actually get the Nobel Prize until about 5 or 10 years after. And so even if they probably recognize that she promised to go back. But that's kind of the rules that just this particular circumstance, but it makes the situation. So I just wanted to talk about that. I know people have had some questions about that on the quiz.
Now,
We talked a lot about things called, I think called the central dogma of biology. And ultimately, the big picture item I want you to take away from this is 2-step process, right? You're taking DNA and you're transcribing it into an RNA molecule. You're packaging that RNA molecule out and sending it out to the rest of the cell, or just shooting it out to the cell or program it. It translated that into protein. You need to both describe transcription and translation in that answer. It can't just be one or the other. Does that make sense? Ultimately, don't be surprised if you see at least one question on this on the exam. We're talking about going from what is the central dogma of biology, and if you don't see both transcription and translation in other shape or form, whether it is definition of those things, or just talking about how DNA RNA and protein. If you don't see both of those things there, it's not a central dogma of biology. It's just training. Does that make sense?
let's talk about mitosis versus meiosis.
First things first. What involves creating haploid cells with meiosis, right? How many cells are produced in mitosis? 2. How many are produced in meiosis? 4. Meiosis, remember, goes from a single cell because of those. So you have not any problem. Break those things apart during step one of this. A is gonna hold on to chromosome B is gonna. And then during that second step of meiosis, you can split those sister chromosomes. But ultimately, you go from 1 to 4. Mitosis, it's a little bit easier. It's simply going from 1 to 2. And it's differently splitting duplication, not worrying about splitting up. As a result, you don't have to do things like you don't have to worry about independent chromosome, that kind of fun stuff. So, straightforward, right? And we'll talk about each individual steps here in just a second. But does anyone have any questions? One thing I do want to highlight is remember that meiosis is pretty much for generating those haploid gamete cells. So that's when you're getting ready to reproduce. And pretty much reproduce away. Now, mind you, that reproduction is still involved with just being asexual. Things like the lizards we talked about, and the snakes, and that sort of thing, when they go part of genesis. They still have a typical meiosis process where they don't have cells that can have to do something. But for the most part, meiosis is primarily just reproduction. Mitosis is more development in organisms, or in something like a bacterial cell or a archaea cell, which is asexual. An individual that's just dominating cells, bacterial and archaea cells can have a line, changing up and mix and matching their genetics and things like that. Now, mind you, they reproduce at such rates that mutation is faster. Just keep in mind, remember, the big mutation generation time, it's not necessarily based on actual physical time is produced. So an organism that produces itself over and has 6 or 7 generations in an hour, it's much more likely to have a higher mutation rate than something that or essentially as a result, organisms live much longer. They may accumulate more mutations in their cells, but they're not necessarily going to produce those.
Let's talk through all of these processes real quick, just one last time.
So it starts in interphase, where basically during mitosis, that cells just kind of sit there, grow, start actually duplicating chromosomes that were. What is it? The chromosomes has the chromosomes. During prophase 1, it's going to condense all those chromosomes back into that nicely packed X shape. Once it's together, it's going to start resolving that into nucleus. You can start to see that there in late prophase, where the spindle fibers are trying to reach towards. Now, during metaphase, all of those chromosomes are going to line up right in the middle, with spindle fibers attaching to them and pulling towards the outside. And again, because this is mitosis and not meiosis, it's going to separate all the sister chromatids during metaphase 1, where it's actually going to pull them apart from each other. So each one of these now has an identical copy of all the DNA in the cell that's still with it. Then finally, during telophase, you're going to start seeing the condensing of both of these new nuclei back together, and the spindle fibers are still going to be pulling apart to start forming that cleavage spur that occurs between cells to what's becoming two cells. And finally, during cytokinesis, those two cells are going to break apart from each other and go from officially one cell to 2 cells. It might have had still nuclei at telophase. That makes sense.
I had a couple of people ask about this, and I just don't think they're in person today. So, or hopefully that made a little bit more sense. I'll just pretend it's a second. All right, let's talk about meiosis.
But meiosis again is very similar. So during meiosis, again, you're going to do the chromosomes right before you get ready to start to end up with those methods. That's how it can break chromosomes together. So starting in prophase 1 with homologous chromosomes, which again are chromosomes that share the same genes, but they don't necessarily the same genes. So they then have, say, the dominant version of allele one and the recessive version of the other. Well, during that process, it's usually during that prophase one, is when you're going to start seeing things that crossing over occur, because the parent has a need that nucleus. And then finally, during metaphase one, all those homologous pairs are going to line up in the middle of the cell. One thing to be very clear here is that crossing over is actually occurring during that prophase stage when they're binding together, not during that phase when they're already staying in the cell. That is just simply the chromosomal line up. And keep in mind too, release, there's a little bit of gray area. I mean, they're kind of all continuum people, where it's like you have late protein versus early protein. So it's kind of hard to always defend and say these ones are exclusively in proteins. Does that make sense? As a whole biology, there's a lot of gray area. There's always an exception to the rule. So after metaphase one, and you break into anaphase one, those two homologous chromosomes are going to be separated, and this is where you're going from that diploid cell to the haploid cell. And then they're going to go through telophase and cytokinesis, where those two cells are going to break apart. The nucleus is going to reform, and they're going to get ready to then divide again. Now, during prophase two, the nucleus is then going to be broken down, and keep in mind, it's only this is a haploid cell. It only has one copy of that homologous chromosome. That haploid, or that during metaphase one or two, sorry, that same chromosome is going to line up in the center. But notice you can see where there's some property over that occur, all that kind of stuff. So it's not exactly like based off of the parental cell. During anaphase two, those two sister chromatids are going to be pulled apart, and then ultimately during telophase and cytokinesis two, you're going to end up with four granddaughter cells, if you will, that are all haploid and have one sister chromatid of that one homologous chromosome. Just for my own sake, what is a homologous chromosome? Can somebody answer? Which of these is a homologous set of homologous chromosomes? One or two? One, right? Where are the sister chromatids being divided? One or two? Two. What's the difference between them? Which ones are the most similar? The chromatids, right? Because they're the identical copies of each other. Which ones aren't an identical copy but have the same genes? Homologous chromosomes, right?
So make sure you keep those two things separated in your mind. They're really easy to confuse. And so you want to make sure that you're making sure that you're talking about either a homologous chromosome or a sister chromatid. Yeah. Yes. So this one combines them into two plots. So instead of showing you where they're pulling apart and the two separated completely, this is just kind of showing.
So what would the prophase two?
So prophase two would be still like two cells with the same homologous chromosome that each has two sister chromatids. All right. Let's practice some Punnett squares. I want to do this with just a couple, like one or two, just to kind of demonstrate some things real fast. Since we have the dot cam actually working today. The homozygous version of the two beta alleles, it's going to always be that particular thing. The heterozygous version, we have one beta allele and one little allele. It'll still be that dominant version, and finally with two little versions. I'm talking about A, dominant. So let's say we're going to albinism instead. That's interesting. So let's say we have two heterozygous individuals.
So here's mom. And dad. And because they're heterozygous, they're both big A, little a. Let's talk about this. How are these? You're gonna have basically a 25% to 50% of each one of these types. So for heterozygous or for homozygous dominant, individual like that. You're then going to do that a little bit here and maybe here. In the way, same thing back here. And finally, the leg, the leg. Does everybody understand why that's that? Okay. Which means that we have big A, little a, little a. Now, for the genotypic expression, which basically means that the ratio of these are 25%. Right? That's you. Finally, 1 of 4. That makes sense. For the phenotype ratio, you have 3 organisms that are going to have that dominant phenotype, right? You have 1 big A, and 2 big A, right? So, for the dominant, 3 out of 4. One out of 4. Looking to agree types that are homozygous for a particular trait. You have a 25% chance you're going to get that recessive trait actually coming through. We have a 50% chance to have 2 more. Another 25% you're going to get. That makes sense. Okay. So let's do one that's a little bit different. Let's say you have a heterozygous recessive. We're going to sort of have let's say we want the percentage for big A and the I'm sorry. What's going to be here? Perfect. Down here. Here. Listen up. All right. Perfect. Same one, right? This one? Perfect. All right. So we know the percentage, which is so we'll do half. Times it by how many individuals? That's 2 out of 4. So again, half. 4. You have 1/4 chance for 25% of getting that combination. Does that all make sense? Does it even make any difference?
See, I promise it's not too bad. And those are probably the most complicated forms you're going to have, honestly, just because, you know, this is an intro level non-majors bio class. I don't expect y'all to know everything.
And most of them have to be like, where you have 2 of them at once, and it'll be like, that's really. And yeah, I'm not gonna accurate for it. Basically, I'll try to keep it straight. Because you only have to be minutes, they're gonna be kind of. As a reminder though, remember you are allowed to write a test. It might not be a bad idea just to like fill it out when you're writing them and on top of it. Because in case something God forbid happens, in the building that they process the scan content blows up or something, it's probably not a bad idea to have things to do. Just in case. No, just like the last two times you did, there will be here. Again, I'm gonna find ways to.
Now, one thing I do want to mention before we move on from Punnett squares is remember there's more than just dominant recessive. You can have X-linked traits where it's based off of what kind of chromosome. So if you've got a chromosome from mom versus chromosomes from dad, it's going to be very different. You know, where you've got an X and Y and X and X, it's going to radically change things. Those forming. Also, with things like codominant, even though the phenotypes will be exactly the same, you can have the phenotypic ratios be different, either for codominant or incomplete dominant. So remember, an incomplete dominant is a situation where either the dominant form is a red flower, the recessive form is a white flower, so the heterozygous version is going to be a pink because it's got half the amount of that particular set of full amount. Now, the codominant situation would be something like blood type, where you could either be A, B, or O, or A, B together. Or A, B, and both codominant. So if you have one of them, you can. Does that make sense? Yes. Yes, you are more than welcome to participate in that. One thing I would recommend is trying to keep and in view of the actual like webcam, and as you know, it goes through that whole process where it asks you to show. More than likely, I'm going to just go ahead and benefit of the doubt, because honestly, this class isn't that hard to cheat in here, because it's part of that. It'd be more detrimental to you to try to cheat in it. Just take a test, any other questions before we move on to this? Now, one thing I do want to mention here is that alternation of generation thing. So remember, in plants, they have a very weird kind of life cycle.
We have a sporophyte and a gametophyte. Now, with those gametophytes, you're going to have a haploid organism, whereas with that sporophyte, you're going to have a diploid organism, or potentially or octoploids in some cases where you have 80 different copies of a chromosome for whatever fucking reason. That's stupid. But ultimately, what happens is that sporophyte is going to be the actual one that's undergoing meiosis and creating those haploid spores. Those haploid spores are going to ultimately grow into those gametophytes. Those gametophytes are going to be the ones that produce the gametes that are then going to be fertilized and turned back into the zygote. Does that make sense? This is going to be a pain. I'm going to warn you now. And the big thing you want to keep in mind with this is knowing which kinds of plants have a dominant sporophyte or gametophyte. So I was asked you, talking about bryophytes, things like mosses, which one is dominant, gametophyte or sporophyte? Gametophyte, right? Because all the little moss you see to grow is the gametophyte itself, and those little stalks only grow. What's in the contrary? Well, what is the contrary? Right.
So which one is it? Gametophyte or sporophyte dominant? Sporophyte, right?
This is only going to show that gametophyte is basically that little ring. Almost. What is the one that's about 50-50%? About half the time it's sporophyte, half the time it's gametophyte. The vascular non-seeded plants, which I know doesn't have a catchy name, which is but colloquial Have any questions about alternation of impressions?
I had one thing as related to the burn,
Mhm.
when I was doing that biology. Non-doctor, either plan was not on there, so I heard.
I would just, so keep in mind too, like as you're doing the identification on iNaturalist, that's separate from what you're gonna actually turn in. So if you find a fern, just call it a fern or a non-Caesar vascular plant, that's fine too. So on the actual document, just write non-seeded vascular plant. Just because there's not really a good colloquial term for that. All right. Now, I did want to kind of run through this again one last time and how these things break apart. So remember, we started with those ancestral produce back here that were things like algae and all that kind of stuff that were completely just thrown into the water. And then they developed that chemical, a lot of them become land plants. So that broke off into, that started our most primitive version of plants, which are those bryophytes, which again have that dominant hermaphroditic state. They don't have any vascular size in them. They don't have any roots. They're just kind of there, and they're reliant on water for things like reducing. And that's all they don't exactly very big, right? They're kind of stuck on the side of the rocks and as high as they're yeah. When you start developing the vascular system, allows to move around things like sugars, waters, all that kind of stuff. That's where you're going to break off into that seedless vascular plant, losses, things like that. So they don't have to seed yet, but they're still needing to rely on water production, but they actually have to be able to move stuff around. And as a result, we see some pretty big birds out there, right? I mean, we're off between, I think it's an olive community earlier, where it's got all of these massive piles of solar plants right there on some of those. Everyone else wants to do it. We're trying to see the development of all of its needs. Creation of those needs. That's going to be your conifers, or ginkgos, or cycads, but for all of your purposes, there's all kinds of trees. And finally, we see the evolution of large and furry bodies. Obviously, so naturally diversified, over 95% all plant life on Earth. It's all because of the proliferation of fruits. I forgot something. I think I saw at least 5 different kinds of plants along. 10? 15? Yeah, you probably saw more than that, right? There's still more out there that you weren't, and it's all those little tiny things, like the little bright blue flowers or the absolutely massive lilies that you saw out there. The firebush where it's like all dark red because of all these little flowers coming up called the fluorescence. Same thing with goldenrods and golden slender tops, all these kinds of things that I'm sure you saw in one way, shape, or form. But does that taxonomy make a little bit more sense now? Just keep in mind, those big differences are what's going to define these groups. And as long as you can define which one has these things and which one doesn't, it'll make life a lot easier. Very similar kind of situation with animals, where you've got development tissue that's going to separate things like sponges away from everything else. Remember, sponges just have actual tissue. Then you're going to get radial symmetry, which is going to create things like Cnidarians versus that bilateral symmetry, which is going to be most of the rest of the organisms that we're going to see here. That's going to be then split into either protostomes or deuterostomes. Remember, protostomes are the ones that develop from the mouth first, where deuterostomes go backwards. And with those deuterostomes, that's where you're going to start seeing things like echinoderms and chordates. Whereas in protostomes, you're going to either get the creatures that can molt themselves or just the weird lump group. So things like arthropods and roundworms are going to be closely related, where roundworms don't have that true coelom. They have that pseudocoelom where it's kind of being formed out of the endoderm. It's not actually coming from mesoderm. Whereas arthropods are going to have all those jointed appendages that make them rather distinct. Then you're going to have this weird group of things like the segmented worms, so your earthworms and a lot of those kinds of things, where they do have a true coelom. Same thing for mollusks. Then flatworms, which is probably the most primitive group of this set. It's going to have just a no true coelom whatsoever. We're just going to have like the two germinal situation. And then finally, remember again, chordates gets a lot more further split beyond that. But take a moment to just, read through these and make sure you understand what the difference between a bilateral versus radial versus asymmetrical is. What does specialization mean? And we're going to have it because that is really important. Things like a coelom, which ones do have it, which ones don't, and which ones have the weird in between. Where absolutely, one could almost call that convergent evolution, if you will. How their digestive tract forms, whether it's incomplete or complete. And finally, if they do have segmentation. Which, ironically, it's kind of weird to think about, but humans have segmentation too, right? Vertebrates in general have some sort of segmentation. It's based off of how our body is shaped. All right. With that being said, does anybody have any other questions that we can talk about for the rest of, about like the 3 minutes that we have left? I hope that this unit was If nobody has any questions, at least in the meantime, just remember our typical rundown of things. Quiz 11 is due on Sunday. Your next exam is Friday. Extra credit is also due on Friday. And then your connecting with biology assignment is due tonight at 11:59 p.m.